Our second presenter today is Michelle Washko of Respana Therapeutics. Michelle?
Thank you, Erica. My name is Michelle Washko, and I'm representing Respana Therapeutics. Respana is developing novel therapeutics for respiratory conditions. The problem that we're attempting to address is influenza-related morbidity, mortality, and negative economic impact. In the US, the direct economic burden of influenza exceeds $10 billion per year, and the combined direct and indirect costs exceed $87 billion per year. The strong human toll can be found in the table at the right. The opportunity we have is to introduce a much needed addition to the therapeutic arsenal. Respana is developing a monoclonal antibody for complications arising from respiratory infection. The initial target is hospitalized influenza patients at high risk. We would then proceed to adults of average risk. The secondary target is patients in ambulatory settings. The total addressable market for these two segments in the US alone is estimated at between $150 and $350 million per year. We see additional opportunities in prevention and emerging coronaviruses. Collectively, the combined opportunity exceeds $2 billion. The technological foundation is a monoclonal antibody specific to surfactant protein A receptor SPR210. This is not pathogen specific, so not susceptible to resistance due to pathogen mutation. We target the host, that is the patient, rather than the virus. We address the maladaptive autoinflammatory response, but importantly, we do not target an individual cytokine pathway. In short, the mAb recalibrates the immune response from inflammatory to reparative. Proof of concept studies show that influenza A infected mice treated with Respana's mAb recover more quickly, have better post-infection lung function, and show better lung histopathology than untreated mice. The competition. There are two antivirals indicated for treatment of influenza. Baloxavir and M2 inhibitors are also used, but infrequently in the US. Antibiotics are typically reserved for special use cases. There are several mAbs either in development or on the marketplace, but none of the approved mAbs are indicated for flu. And importantly, again, Respana's antibody does not target individual cytokine pathways like these. We have a very strong team, both on the scientific side and on the management side, and we are currently in negotiations to onboard our first two scientific advisory board members. Here you see the commercialization plan laid out for you quarter by quarter, culminating in an IND filing anticipated in Q1 of 2023. If we were to be awarded the funds, we would use it for comprehensive analysis of the lead humanized monoclonal variants and for robust IP prosecution in both Europe and Australia. In short, the case for Respana is a strong one. We have a compelling value proposition for both patients and the healthcare ecosystem. We have a unique target with highly promising preclinical data. The IP position is strong. The scientific and management teams are both also very strong, and we have early external validation. And with that, I'll leave it to questions. Thank you.
Thank you, Michelle. Nice presentation. Okay, so we will begin the Q&A, starting with Alana.
Hi, Michelle. That was very nice. Thank you, and very interesting. You talked about a large market size there. And when you're thinking about your total addressable market of 150 to 350 million, I'm curious, what % of that total addressable market is actually your specific solution? Can you actually address sort of your service addressable market? And how did you get to those numbers?
So those data were driven largely by a paper that laid out the costs. Actually, if we go back to this, can I go back to my slides and show you the second slide there? That might be helpful. We'll share that. So if you look at the chart here on the right, this is data that comes from, this is sort of a compilation of data from a variety of sources, primarily in that particular chart, the CDC, but there's a lot of literature on influenza out there. And we've looked at some papers that look at the total economic burden of influenza, particularly one, I can't quite remember the author's name, but it's cited in the one pager that's part of the packet. And that particular paper looks at average influenza costs over a 10-year period. And what we did is look at the costs for both inpatient and outpatient folks, and we looked at the total economic burden on average, and we looked at, we combined that, we triangulated with the data for the antivirals that I referenced and what their market share is and what they're bringing in annual sales, and that's how we arrived at 150 to 350 million.
you.
Sure.
Okay, Bruce, do you have any questions?
Yes, Michelle, very nice presentation. Thank you. A few quick things. One is, have you had any preliminary discussions with FDA about, clinical trial requirements and your regulatory pathway?
We have not gotten there yet, no. We have been consulting with various experts in the field. There were a bunch of different pathways that we could take here. We do think, based on these early discussions, that we would start with inpatient folks in the beginning with high-risk adults, those over 55, those who already have underlying conditions. But there are certainly other methodologies, and for that, we are looking to consult with additional clinicians. We do have Dr. Haverly-Lack, who is a Penn State person, Penn State faculty and clinician who was advising us, and also the SAB members who would be coming on board also have particular expertise in that area. So the short answer to your question is no, we've not had preliminary discussions with FDA. We know there are lots of options here, and we know that we need to do additional research with folks who are actively in the clinic to understand exactly what our opportunities are there and what the appropriate pathway would be.
Okay. And how much cash do you estimate it'll take you to get to your IND filing, what you outlined in your Gantt chart?
To get to the IND, we think that probably we need about, I would say, between $1.5 million and $2 million, probably get to that IND filing.
Okay.
A lot of that would be in the cost, right? As I think many of you and around the room today would understand, is that to have the material made in the quantity that's sufficient for a clinical trial is going to be the biggest lift there. That could be as much as $1 million. We don't really have a visibility on that yet. It could be appreciably less, but we don't know that yet.
And are you confident it is manufacturable at some reasonable scale?
There's no reason to think that it won't be. The early affinity data looks good. So we feel like it's probably not going to be huge amounts of material. But again, that remains to be seen until we consult more with drug formulation folks and explore those into further detail with CROs. We have been in discussions with CROs who have reputations in this space, but I would be exaggerating if I said we have strong visibility on that just yet.
Thank you.
Mm-hmm.
Hi, Michelle. Thanks for that presentation. I have a question about the discovery process. So how did you originally come up with this indication? Was it you already had this indication and you were screening for it, or did you discover the antibody first and then try and look for an indication later?
So I'll do my best to answer that accurately, but that's a question that's perhaps best reserved for our scientific founders, Drs. Kronios and Christensen. My understanding is that he identified the target first, the surfactant protein A. That is his discovery, that SPR210. And his early work was all with influenza. So what I haven't stressed here in this presentation is that it looks like we have opportunities in lots of areas of the respiratory space, but all the early work has been done with influenza. There is some, it's mostly viral, some bacterial influenza as well.
So you were hinting at potential other indications.
Yes, so there's every reason to believe that we could attack some other coronaviruses for sure. Prevention, again, theoretically, looks like it should be entirely plausible. We also think because of the reparative nature of the MAB, we believe that we might even have applications in fibrotic diseases. And actually, we've explored that in conversations with some potential SAB members with whom we've had discussions. And that seems to be of great interest to those folks as well.
All right, thanks. That's it for me.
Okay. Thank you, judges, for your questions. Michelle, great presentation. Thank you.